This topic is about the process of microsporogenesis and microgametogenesis. So, what is microsporogenesis? Microsporogenesis is the formation of microspores inside the microsporangium by mitotic or reduction division. The formation of pollen grains from the microspore is termed as the microgametogenesis. Now, we see about the formation of microspores. The developing anther consists of two types of cells. One is reproductive and another one is non-reproductive cells. The diploid cells are the reproductive cell in the sporogenous tissue of microsporangium called meiocytes or pollen mother cells. Divide by the meiosis to produce four haploid microspores. The first reduction is divide gives two nuclei and the second division finally gives the tetrahedra of microspores. And the last one, the non-reproductive cells from the anther tissue layer including epidermal, tapetal and cortical layer. The tapetal layer functions as the nutritive tissue of the developing anther. Now we see about the formation of pollen grains. The formation of pollen grains from the microspores is termed as the microgametogenesis. It begins with the expansion of the microspores and the formation of the large vacuole. It is followed by displacement of the microspore nucleus from the center to one side of the cell against the microspore wall. Here, the first pollen mitosis takes place that produce a large vegetative cell and the small generative cell containing the haploid nucleus. The generative cell that has attached to the microspore wall starts detaching and is engulfed by the vegetative cell forming a NQ cell within the cell structure. The engulfed vegetative cells that undergo pollen mitosis to form two pollen sperm cell packed in the cytoplasm. This is the two stages formation of pollen grains from microsporogenesis and microgametogenesis. Next, we are going to see about the gametogenesis in plants. Gametogenesis is a biological process by which haploid male and female gametes are formed. This occurs in both plant and animals. In higher plants, there are two stages that are evolved: sporogenesis and gametogenesis. Sporogenesis is formation of spores, whereas the gametogenesis is the formation of gametes. And next is about microgametogenesis, the process of development of male gametes in the microspore or pollen grain is termed as the microgametogenesis. Microspores symbolize the initiation of the male gametophytic generation. During gametogenesis, the pollen nucleus divides mitotically to yield the unequal cells. One, the large one is vegetative cell or tube cell which forms the pollen tube, and the smaller one situated towards the wall is generative cells which again divides by mitosis to form two sperms, that is male gametes. After germination of pollen grains, the generative cell may divide inside the pollen grain or in the pollen tube. The generative cell divides by the mitosis division. If this division take place when they are anther, the pollen are shed three cell stages. If it take place after the pollen germination, they will shed at two cell stages. The exine ruptures and the intine forms the pollen tube. The vegetative nucleus enters the pollen tube. Now it is known as the tube nucleus. The generative cells give rise to the two male nuclei, whereas the vegetative cells gives rise to the pollen tube. This explains the process of microgametogenesis and microsporogenesis in the plant of